Thank you, Dr. Fries, and thanks for giving us the opportunity to present our work in this important annual Rare Disease Day. It's very important to emphasize research findings on rare diseases and therapies. They may not only help patients with rare diseases, they may also teach us new insights into more common disorders and perhaps find therapies for more common disorders. So I will give an example of what Eric calls his third domain, understanding the biology of disease. And this may help answer the question about finding a mutation in, like, what kind of research is involved in finding a mutation for a patient with a rare disease and to bring it to therapy. And I'll only give you an example of one rare disease, and there are so many rare diseases out there, so this will really take decades to solve for many other for the whole variety of disorders. See how I work this. So today I'll talk on our research findings and efforts to bring to human clinical trial N-acetylmannosamine, ManNAc, or Sialic acid as therapy for disorders of hyposialylation. I'll first start out with some more basic research and get to the therapy later on. So hang in there in the beginning. How do I move this forward?
Use keyboard.
This is not mine.
Keyboard, which is there.
Oh. Yeah, I got it.
Got it?
Yeah.
All right.
I'll talk about disorders of hyposialylation. First, a little background about sialic acid itself. It's a negatively charged acidic sugar, and it's a terminal sugar on glycans. And mostly due to its negative charge, it functions in both normal physiological processes, such as cell-cell interaction, signal transduction, embryogenesis, but it also functions in abnormal pathological processes, such as microbe binding, inflammation, immune responses, and cancer. The most common sialic acid is N-acetylmannosaminic acid, or N5AC. This is the precursor for all other sialic acids, and it's commonly referred to as just sialic acid. Here is a sialic acid synthesis pathway in the cell. It starts out really with glucose, which can undergo many conversions, but it can also become UDP-glucNAc. And then the first really committed step for sialic acid synthesis, which means there's no way back, is the conversion of UDP-glucNAc to ManNAc and ManNAc to ManNAc 6-phosphate. This is mediated by a complex bifunctional enzyme, which is called UDP-glucNAc 2 epimerase, ManNAc kinase, which the rest of my talk is about. Further conversion to sialic acid in this pathway is the conversion ManNAc 6-phosphate, 2 more steps to sialic acid. Sialic acid is then, for still unknown reasons, shuttled through the nucleus to become charged CMP sialic acid, which is able to feedback inhibit this very first committed step of sialic acid synthesis in the allosteric site of this enzyme, thereby creating a very tight feedback regulation loop, a very tight regulation of the amount of free sialic acid present in the cell. CMP sialic acid is used by the Golgi to sialylate glycans, which can then perform their functions, like they're mostly membrane associated components, and glycans get turned over in lysosomes where free sialic acid can be released for possible reuse. Mutations in this bifunctional enzyme cause decreased amounts of sialic acid produced and can result in hyposialylation. Mutations in this enzyme causes the rare disease HIBM. HIBM stands for Hereditary Inclusion Body Myopathy, which is an adult onset progressive muscular disorder. The onset is in the 20s to mid-30s. Most patients become wheelchair-bound within 10 to 20 years after onset. It initially presents with proximal features like foot drop and finger weakness, but eventually progresses to more severe weakness and atrophy of many proximal and distal muscle groups, but it relatively spares certain muscles, including the quadriceps. You see foot drop here, and you see a relative quadriceps sparing here. And the quadriceps sparing you can see on an upper tight MRI here. The dark is the quadriceps, while the hamstring is almost completely wasted. Patients' mental cranial nerve sensation coordination are normal. Creatine kinase is surprisingly normal to very slightly elevated. The clinical features, even though the exact pathologic mechanism is unknown, are most likely due to hyposialylation because the enzyme defect the gene defect is known. There are about 1,000, it's estimated, HIBM patients worldwide. There exists a Persian Jewish and Japanese isolate. And since the identification of Gne mutations with HIBM, around in 2001 the gene was identified, there are over 80 different Gne mutations described so far. And most of them are missense because you need some residual Gne activity, you need some sialic acid to be compatible with life. To further explore the HIBM pathology and to test possible therapies, several mouse models for HIBM have been created. A Gne complete knockout is embryonic lethal. You cannot live without Gne, you cannot live without sialic acid. A transgenic mouse has been created mimicking the Japanese founder mutation of this disease. And this mouse mimicked the adult onset muscular features of the disease. And we created a knock-in model through homologous recombination mimicking the Persian Jewish founder mutation of HIBM, about which I'll show you a few results. So our initial matings of heterozygous, like two carrier mice, resulted in 101 pups, but to our surprise, only one mutant pup survived beyond weaning, that is postnatal day 21, and we found that all mutant pups, most, died before day 3 of life, which was very surprising because the disease is an adult onset muscular dystrophy. We found in utero a Mendelian distribution, and which indicated that most mutant mice died between birth and day 3 of life. At day 2, the mutant mice were a little smaller than their litter mates. Their stomachs did contain milk, indicating that they were drinking. So we then investigated if perhaps their muscle phenotype was similar to the human disease. We performed several studies, including on the dystrophin glycoprotein complex, but at this age, day 2 of life, we could not find any abnormalities in their muscle histology. But we saw that their kidneys were severely affected. Mutant mice had these hemorrhagic kidneys, cystic dilatations in the cortex, proteinaceous debris and red cell infiltrates in the tubules and the glomeruli, and the mice had severe proteinuria. This is an EM of a normal mouse glomerulus, in which you see the glomerular lumen here. That is where the blood flows through, and it gets filtered through the through the glomerular basement membrane and some cells that I'll talk about into the pre-urine. This filtration complex consists of endothelial cells, a basement membrane, and these so-called podocyte foot processes, these finger-like structures, and there are these filtration slits in between. These podocyte foot processes and filtration slits contain highly glycosylated and thus sialylated glycans. And it's believed that the negative charge on these sialic acids help maintain the podocyte shape and help keep these filtration slits open. And it's been shown that if you clip the sialic acids off in like a rat model, the podocytes fuse and flatten because the loss of charge. The two most abundant glycans in this complex is podocalixin in the podocyte foot process membrane and nephrin in the slit diaphragm membrane. And they're both glycosylated. Then we looked at the kidney structure of our mutant mice, and we saw that these podocyte foot processes were mostly fused and flattened, shown here. Only a few filtration slits remained, and they formed these tight junctions. There was not a real good filtration slit there. So then we checked these kidneys, podocalixin and nephrin, sialylation status by Western blot. So you see here for podocalixin in normal and heterozygous mice, it shows this large blob around 150 kilodaltons. If we treat with neuraminidase, which clips off sialic acid groups from proteins, this band shifts up because it's less negatively charged. And then if you compare this with mutant mice kidneys, they have this upper band naturally, indicating hyposialylation of podocalixin in these membranes. For nephrin, the same thing, the wild type and heterozygous mice treated with neuraminidase, the band shifts down, and that is because a difference in molecular weight. And the mutant mice had this lower band naturally, indicating hyposialylated nephrin. So hyposialylated nephrin and podocalixin and likely other glycans most likely caused the flattening and fusing of this podocyte, the podocytopathy in this mouse model. You can also see that the basement membrane had segmental splitting, which is a feature of some human kidney disorders as well. And then the proteinuria of the mice, you see here it was mostly albumin that was excreted, indicating a glomerular defect over a tubular defect. You see here mutant mice, albumin compared to wild type and heterozygous. And another way to show hyposialylation, we showed it here in kidney for our mouse model, but you can potentially show it this way in any other tissue or for any other disease, is by a panel of lectins. Lectins bind to specific glycan structures. For example, here the lectins WGA and SNA bind end group sialic acids. And you see here in mutant mice, in wild type mice, there's a bright stain. There's enough sialic acid in the glomeruli of kidneys, while in mutant mice, these two lectins stain less, indicating hyposialylation. Then these other four lectins only bind to a glycan if the sialic acid is not present, so bind free galactoses. And the other two bind O-linked, bind if sialic acid is absent to O-linked glycans only. And you see for all these four lectins, the mutant mice stain the glomeruli stain bright, but the wild type mice, it's hardly present, again indicating hyposialylation. Now, what would be a possible treatment for these mice and eventually patients with HIBM? Well, since there is a mutation in GNE, these two enzymes, the first, and therefore hyposialylation, the first thing that would come to mind is to give the patients free sialic acid, to add sialic acid to their system. However, sialic acid has a negative charge and may be hard to get into cells. So a second option would be to give sialylated glycans, which have a lot of sialic acid that can be turned over by the cell, and the sialic acid that is released can be used for glycans that need it more. In fact, we performed a clinical trial on 4 HIBM patients in 2006, published in 2007, and by treating them with intravenous immune globulins, IVIG, which is highly sialylated. We gave them 3 or 4 different treatments. The outcomes were encouraging, but we could not really find in that the patients got stronger. Their muscle strength improved a little, but we could not find a very good biochemical outcome parameter at that time. Plus an IVIG treatment every month becomes very expensive and too demanding on patients too. But bound sialic acid may be a therapy to treat hyposialylation. And then the third thing a third treatment that could be possible is treatment with ManNAc and N-acetylmannosamine, which is the only uncharged sugar in this pathway. It's a physiological sugar, and it comes into this pathway after the feedback inhibition step. So it is totally it produces sialic acid independent of the feedback inhibition. Now you may ask, but what if patients have mutations in the kinase here and you feed ManNAc? Well, there are other kinases in the cell, in particular GlucNAc kinase, that can convert ManNAc to ManNAc 6-phosphate. So ManNAc would be a potential therapy to treat hyposialylation, and especially because it's a physiological sugar and it's uncharged. So we fed pregnant female mice of our HIBM mouse model sialic acid or ManNAc in their drinking water. And we assessed how many mutant mice survived beyond these 3 days of life. They would die before 3 days if you would not treat them. And we found that sialic acid feeding of 1 gram per kilogram per day did not yield significantly higher amount of mutant survivors. But ManNAc feeding resulted in about 50% of mutant mice surviving beyond day 3. These mice were taken off ManNAc after weaning, after day 21, and they continued to live. They remained smaller than their litter mates, and looking at their kidney histology, it improved. I'll show you in the next slide. We measured increased Gne enzyme activities, and we showed with lectins improved sialylation of glycans. And importantly, when these mice were around 4, 5, 6 months old, we found hyposialylation of their muscles, mimicking the human, potentially getting into similar pathology as the human HIBM disease. So we published all this work in 2007. And we were very excited. We thought we could give ManNAc to humans, and I'll come back to that later. We're still trying to get it to humans. So here's some of the outcomes that we saw in our mice. So the mice that survived after orally feeding ManNAc, we saw that their podocyte food processes came back, not as nice as in normals, but they were there, and their new filtration slits appeared. So the glomerular structure improved. Also, the basement membrane was not split anymore. ManNAc upshifted when untreated compared to normal. It shifted down after treatment, indicating resialylation of podocalixin. And the same for nephrin. Before treatment, untreated, you see this downshift, and after treatment, it came back to normal, indicating resialylation of nephrin upon ManNAc feeding. And also the albuminuria, the proteinuria, that was pretty high here in the mutant mice, decreased. And the lectin stainings, so before there was not much staining of sialic acid end groups, and after treatment, the stain there was more intensity of the staining, and the 4 lectins that show hyposialylation stained brightly here improved somewhat. There remains hyposialylation in the treated mice. So we are looking for we're mimicking a muscular dystrophy mice, but unexpectedly we found this kidney phenotype. So that brought up the question, are there human glomerular diseases due to hyposialylation? And if so, if they exist, can they be treated with ManNAc? So in fact, in the literature, there are some unexplained forms of minimal change nephropathy, focal segmental glomerular sclerosis, and other unexplained forms of idiopathic nephrotic syndrome where hyposialylation is indicated, either with a lectin panel or with an upshift of podocalyxin. So I guess the future will tell if these glomerular diseases can be potentially treated by ManNAc too, even though the exact molecular basis of these diseases is not known. So it may not be possible to treat them with ManNAc. So then, as I said, we found that ManNAc could increase sialylation in our mouse model around 2006. In May 2007, NIH Technology Transfer Office acquired a patent, N-acetylmannosamine as a therapeutic agent. In May 2007, New Zealand Pharmaceuticals agreed to provide ManNAc, GLP. A clinical protocol was written and approved by our institute's scientific review committee and later by the institute's review board. Our paper was published. The NIH Pharmaceutical Development Service was on board to start helping with bringing ManNAc to patients in a clinical protocol. An investigational new drug application was submitted to the FDA, and 29 days, and actually patients were contacted and everyone was enthusiastic, and 29 days after the application, the FDA put the protocol on a full clinical hold, requiring the whole set of toxicity and preclinical studies. At that time, it was estimated to be about $1.2 million. It is more expensive now. It has gotten more expensive over the years since 2007. So we attempted between 2007 and 2010 different avenues to try to raise money, to try to contact patient advocacy group, other grants, other rare disease treatment initiatives. The whole process was pretty frustrating, and we could not get enough money. Some talks with the FDA happened. We really needed to do the toxicity studies. And in 2010, NIH launched a new program, the so-called TREND program, the Therapies for Rare and Neglected Diseases. They chose a few pilot projects, including HIBM. So they are helping with the preclinical studies to bring ManNAc as a treatment for HIBM to patients. In May 2010, New Zealand Pharmaceuticals acquired the license to market ManNAc. And in 2010, again, this process goes really slow. A company was contracted to perform extensive studies on a rodent and a non-rodent model, in our case, a rat and a dog model. And in fact, these studies are finished now. They were finished by the end of last year, and they're in the process of being reported and analyzed and written up, hopefully to provide the FDA with an answer to the clinical hold, hopefully this summer. In the meantime, a natural history study has started at NIH, so we can establish some baseline parameters before treating patients with ManNAc. Unfortunately, many patients, well, maybe fortunately, many patients have heard about this, so there is some off-label use of or ManNAc or other compounds that can increase sialylation. So we may not have a naive population to start with, or for now, the projected start of the human clinical protocol for ManNAc and HIBM is the end of this year. We will see what happens. So in the meantime, since 2007, the excitement that HIBM could be treated with something else than ManNAc got picked up by other research groups and companies too. So in the meantime, free sialic therapy has been explored. A human phase 1 trial has in fact been conducted in 2 different centers, both in Japan and by the company Ultragenics here in California. And both of them are now gearing up for phase 2 trials. Bound sialic acid therapy, as I said, IVIG was tried by us in 2006, 2007. And a mouse model, the Japanese mouse model of HIBM, was treated with oral sialyl lactose. Sialyl lactose is very high in sialic acid. And it was beneficial for the muscle phenotype in this mouse model. So sialyl lactose is very high in mother milk. However, it may be harmful to use cow milk or sialyl lactose isolated from animal milk because the sialylation of sialyl lactose is different than human sialylation. So sialyl lactose therapy would have to be done with perhaps human mother milk sialyl lactose isolated from it. And I think Dr. Vries has pursued this a little, has looked into this a little bit. And then another therapy that has started is GNA gene therapy. The company Gradalis in Texas has inserted the GNA cDNA in a plasmid and inserted this plasmid in liposomes. They call GNA lipoplex. They IV injected, actually they first intramuscular injected one HIBM patient with this plasmid, and later injected the same patient IV with this plasmid. And the patient may have gotten some strength back. It may have resialylated her muscles a little bit. And I think they're gearing up for a trial in more than one patient. So since we have our mouse model, that's where mutant mice, if they do not get sialic acid or a precursor given to them, if they die within 3 days of life. So it's a really good model to test different therapies for the hyposialylation. So if mice are untreated, only 8% of mutant mice will survive. If you give them ManNAc orally, about 48% of mutant mice will survive. If you give them sialic acid orally, only 10% survives. So we worked with this company Gradalis to put ManNAc and sialic acid in liposomes, in their lipoplex. So we injected newborn mice at day 1 with ManNAc lipoplex or sialic acid lipoplex or GNA lipoplex. And we assessed how many mice survived beyond day 3. We found that virtually almost every ManNAc injected mouse survived beyond day 3 of life. The ones that died were really because of our technique of injecting at day 1 the ManNAc lipoplex. Sialic acid lipoplex also increased survival significantly. We think not as good as ManNAc, but it increased it. And it looks like it's really a matter of delivery. Oral sialic acid therapy may not be as efficient as oral ManNAc therapy, but IV, if you put it in a liposome or deliver it straight to the cell, it may have a similar efficiency. GNA gene therapy in our mice, injecting at day 1, did not yield any more survivors. However, the GNA gene may not the gene therapy may not have allowed too much time for these mice to make enough sialic acid, because the gene has to be translated into the protein has to make sialic acid, the sialic acid has to be put on glycans, and this all has to be done within a day or two. So that may be the reason why the gene therapy did not work on our mouse model. So in conclusion, I think I showed how disorders of hyposialylation could potentially be treated by ManNAc, by free sialic acid, or by bound sialic acid. And the disorder HIBM is a very good prototype disorder because the gene defect is known. We know it causes hyposialylation because there's defects in this GNE gene. So this disorder can potentially be a prototype disorder for therapy of other hyposialylation disorders. What could be other disorders of hyposialylation? As I mentioned, renal disorders with proteinuria that have fused podocyte foot processes, which have split basement membranes of unexplained pathology where no gene defects have been associated to yet. Certain cancers are hyposialylated. Would ManNAc help? Or other therapies. Disorders of autophagosomes have been described to be caused by could be caused by hyposialylation. Certain amyloid accumulation disorders could be started by local hyposialylation. Could they be treated with some of these compounds? I hope I can report more on this in next year's Rare Disease Symposium. So thank you for your attention.
Thanks very much. We've just got time for one quick question. Any questions from over here?
Is there an easy screen to tackle sialylation, like would a sialic acid work, or do you need to do
that? Yeah. So the question is there an easy screen for hyposialylation? Would a urinary or serum test help? For HIBM, it is not an easy screen, but for all, because only in HIBM we think only certain glycans are hyposialylated because patients only get a muscle phenotype. Only specific muscle proteins are hyposialylated. So the total sialylation in urine or the total sialylation in serum is the same. For other disorders of sialylation, perhaps it will work. Measuring sialic acid is not standard care. There are no standard assays for serum or urinary sialic acid amounts. So, yeah.
Okay, thanks. Thorsten, it'll take time away from your talk if you want to do it. All right, if your question is important enough.
No, I have just one comment and one short question. The comment is, when Manos was approved in Europe for the treatment of CDG1B, the way we did it, it was approved as a dietary supplement.
Yeah.
And it's distributed by a company that has specialized in dietary products for other metabolism. Yeah. Instead of making a new medication out of it. Yeah. Because after all, it's a sugar that you're getting. Yeah. And so you have less hassle with this.
ManNAc is not an over-the-counter sugar. It's not a dietary supplement. You cannot buy ManNAc in a store.
No, but it's not considered a natural product by the FDA, and that's why they went through all this.
Okay.
Yeah.
And the question is, if you continue to give ManNAc after weaning, you said you discontinued it, does it prevent the development of the hyposialylation disorder in the muscle?
Yes.
It does?
Yes.
Good.
Also, if we inject it in the lipoplex at a later age, it prevents the hyposialylation.